Hey everyone! This video is going to help you with objectives 23 through 25 on diffusion and passive transport. I actually made it last year, and the slides reflect last year's objective numbers, so don't let that throw you. I'm just updating this introductory portion to let you know that although the slides say objectives 22 through 24, they really reflect objectives 23 through 25 in your 2014 packet. So make sure you have your notebook out, turn to those objectives, and get ready to take some notes on anything you may have missed. Thanks! So the reason we're learning about the cell membrane is because it's critically important to how the cell functions. The cell membrane regulates what goes into and out of the cell. It's able to regulate this through both passive and active transport. So this video we're going to be talking about passive transport. And passive transport really is based on the fundamental physics principle of diffusion. So what is diffusion? Diffusion is the tendency for particles to spread out. And this means that their entropy increases. Entropy is a measure of disorder in a system or chaos in a system. And so there is a generally accepted principle that entropy or chaos or disorder increases, and that it takes energy for us to keep things in order. And that is reflected in your bedroom floor, right? Which no matter how hard you try to keep your clothes from ending up on your bedroom floor, they always do. The disorder increases unless you put a tremendous amount of energy into keeping things organized. And the same is true for biological systems. So diffusion is just the tendency for particles to spread out. This means they move from high concentration to low concentration. So if you have a beaker of water and you put some food coloring in it, eventually that will spread out and fill the entire beaker. This is due to random molecular motion or vibration. As you know, molecules are always in motion. And so as they constantly are bumping into one another, they start to move around and fill whatever space is available. So if one side of a membrane has more molecules hitting it than the other side, eventually, all things being equal, those molecules will reach what we call a dynamic equilibrium. Dynamic means in motion, and equilibrium means equal on both sides. A dynamic equilibrium, it's really important to note, does not mean that nothing is moving. It means that the molecules are moving out and in at the same rate. So overall, if you were to look at the net movement, there would be no net movement, but there is movement both into and out of the cell at equal rates. Okay? The other term that we really need to familiarize ourselves with, particularly in this objective, is concentration gradient. Gradient is just a difference in concentration. So I don't know if any of you have seen those t-shirts that go from light color to dark, or yarn that goes from one color to another. Those are color gradients. It's a difference in the concentration of the dye. We talk about concentration gradients across a membrane because when you have a difference in concentration, the molecules are going to want to even that out. They're going to want to spread out into areas of lower concentration. So we say that when molecules do that, that they move down the gradient. And so diffusion is a process of molecules moving down the gradient. They're going from high to low. So passive transport is basically diffusion. There's going to end up being two types of transport across a membrane. Passive, which doesn't require any energy and is essentially diffusion, and active transport, which will require energy, which we're going to learn about later. So passive transport is diffusion down the gradient from high to low and requires no energy. And we call that kind of diffusion simple diffusion. And the kind of molecules that can diffuse across the membrane without any help in the form of simple diffusion are nonpolar or hydrophobic molecules, mostly, because the phospholipid bilayer is mostly nonpolar. And if you remember how likes dissolve likes, you'll know that those lipid tails of the phospholipids are really very accommodating to other lipids. Water also is able to cross the membrane, even though it is not nonpolar, it's polar, but it's a very small molecule, and because of the polar heads of the phospholipids, it is able to get through. There are, however, some molecules that aren't able to get through the membrane very easily. And so if we want them to diffuse across the membrane, they need some help. And so we call that facilitated diffusion. And it still doesn't require any energy, but it's just when the diffusion takes place through a channel or a tunnel protein in the membrane. So they provide sort of a little hole through which molecules can travel that wouldn't otherwise be able to get through. So molecules that applies to are ions, molecules like glucose and amino acids, as well as water. Now, you'll notice that water is in two places on this slide, and that's not a mistake. Water can diffuse across the cell membrane without any help, but there are these proteins called aquaporins that are tunnels basically just for water. So if a cell needs to acquire or get rid of a large amount of water in a short period of time, it can use these facilitated diffusion tunnel proteins to help that process occur with greater efficiency. And we're going to talk a little bit later about why a cell might need to get rid of or obtain large amounts of water. The third term here that we should definitely make note of is osmosis. Osmosis is sort of under the umbrella term of diffusion, and osmosis is specifically diffusion of water. And we're going to learn more about osmosis in Objective 28, but it's just that sometimes a membrane might be permeable to water, but not to other things. So let's say you had a membrane, and on one side was a very sugary solution of water and sugar, and on the other side was just plain water. The sugar might not be able to get through the membrane, so it can't go from high to low because it can't go through, but the water can. And so the water will go from high water concentration to low water concentration. So in actuality, the water will move into the more concentrated solution to try to even that out. So the water is moving down its own gradient, although it appears to be moving up the gradient if you look at the sugar. And I know that's a little bit confusing, but we're going to be practicing a lot with that next week, so don't worry. This is the original U-tube. It's a U-shaped tube that is often used to demonstrate the process of osmosis. And you can see that there is on the left a less concentrated solution and on the right a more concentrated solution. And between them is a selectively permeable membrane. And you can see that over time, because those little dots, whatever the solute is not able to pass through that membrane, the water must pass to try to even out the concentrations. And so what you see is an uneven volume in the U-tube, which is very interesting. So the water actually is going to move over to the right in order to make the concentrations equal. So the dynamic equilibrium in this case means that one level of the U-tube is higher than the other. And we'll talk more about this, like I said, in Objective 28.